Okay. Hi everyone, thank you all for being here. I'm Stefania Pirrotta from the University of Padova, and I will talk about uncovering mitochondrial activity by transcriptome data with mitology. So we're talking about mitochondria. They are critically important organs for the function of the cells, and they are most known to be the powerhouse of the cells, as they produce all the energy in form of ATP for cells to function through the decarboxylic cycle, acid cycle. But they also are involved in many different biological processes, as some of the most famous are the ones reported here: liquid metabolism, calcium homeostasis. So when mitochondria do not work well, they actually is a problem for the cell. And actually, they are involved in many different complex diseases. So when you want to study something like that, usually you will perform a pathway analysis. But actually, all the common databases that we usually use for pathway analysis are not really specific for our mitochondrial activity. So the aim of the project was to try to create something that could allow you to perform large-scale analysis, but focus only on the mitochondrial part. Why? Because many of the pathways in which mitochondria are involved usually are complex pathways that may have a part that actually is outside the mitochondria, and then a part that is inside the mitochondria. So to fill this gap, we developed mitology, our package, that allows you to infer and dissect only what's happening inside the mitochondrial part. So the first step was to collect mitochondrial genes. These are not meant to be only the genes coming from the mitochondrial genome, but also genes whose protein are localized and inside the mitochondria or on its membrane, and also the ancillary genes that are involved in the mitochondrial activities. So to collect the genes, we use the specific mitochondrial-oriented database that are INPI, Mitocarta, and SecDR, and also from the gene ontology from the cellular compartment area. We can see that many genes are shared among the 4 databases, but actually each of them has its own subset of genes. So we decided just to take the union of these 4 lists, and we obtained almost 3,000 genes. Then we have to select the mitochondrial gene set, explaining the different biological functions. Actually, Mitocarta already provide database of pathways. Is like Reactome, but just mitochondria-oriented pathways. They provide 122 pathways, but less than 40% of the genes in our gene list are annotated inside this pathway. So it seems like we are missing something. So we decided to explore also the Reactome and gene ontology databases. There are around 2,000 pathways in Reactome, more than 11,000 terms in gene ontology. What we wanted to do was to filter these pathway in terms and focus just on the mitochondrial relevant processes. So the aim was to minimize the number of gene sets, maximizing the number of genes. So at the end of the filtering, we obtained 83 gene sets from Reactome and 286 gene sets from gene ontology. I'm not using anymore the word pathways and terms because we are not taking the entire pathways. As I said before, from an entire pathway, we're just taking the mitochondrial part. So I'm calling them now just gene sets. Here you can see the final 3 dendron structure from the pathway gene sets selected for the different databases. And with this information, we develop mitology that can take in input bulk, single cell, or special transcriptomic data and perform a single cell gene set analysis when you have just to choose the database you're interested in. And allows you to visualize as circular heat maps the final enrichment scores plotted on the final structure that I showed you before. Just to show you an example, this is a special breast cancer dataset coming from the 10x databases. We asked an anatomopathologist to annotate the area for us, and you can see that in red there is the tumor area that had some necrosis tissue inside, and that was surrounded by calf and lymphocytes area. You can apply hematology on that. For example, here I'm showing you the results obtained with the reactome databases. Just to sum it up, I'm not showing you all the scores obtained for each spot, but I'm taking the median for each annotation type. So you can see that divided by tumor, calf, lymphocytes, blood vessel, necrosis, lymphocytes, and stroma. I don't know if you can read them. And so in this way, you can compare the enrichment scores among them. So for example, for this gene set, mitochondrial and subclass biogenesis, we can see a difference in the enrichment score for tumor, calf, and lymphocytes against the others. Obviously, you can also especially plot the single enrichment scores obtained. So here you can clearly see that for this annotation, for these types, it seems to be more enriched than the other annotation types. So this was just to give you an idea of how you can use the package to explore just the mitochondrial activity on your data. So to conclude, we are developing this new tool to explore mitochondrial in different types of transcriptomic data. It is available on GitHub, but we are still working on it, so it may not work still properly for now. Once it will be finished, we plan also to submit it to BioConductor. Thank you for your attention. Also, I thank all the members of my labs.
So, a bunch of questions? Thanks for the great talk. There were some really, really beautiful visualizations in your slides.
Thank you.
How many of those are actually from your package? Or maybe the sneaky question is, how many of those did you tweak in Illustrator? They were really, really beautiful.
Thank you. For the visualization, here I use Jesus Fabulous package. For this one, yeah, I use the GG3 package. And this is the type of visualization that you can make with my package that include the endogram structures. So every time you can plot your scores on the endogram. Yeah.
I'll make a second question. So is there a particular reason why you're using SSCSA as a gene score out of efficiency or to compare different ones, or is something that you can parameterize in your packets?
No, we're still working on it. Yeah, exactly. This is one of the critical points. We're still working on it. For now, we decided to start with that. But once the structural features of the package will be completely finished and okay, we will also focus on patterns and would be the better enrichment analysis to do, or also maybe just let the user choose different types of enrichment analysis.
Okay, thank you. So let's thank Stefania again. Maryna, Chepeleva. Thank you, Maryna.
Thank you. Hello, everyone. My name is Maryna. I'm a PhD student in Metabolomics Data Science Research Group in Luxembourg Institute of Health. And today I'm happy to present part of my work that I'm doing here. So in cancer department, we're working on understanding processes that cause cancer progression. In my project, focus on integration of transcriptomics and metabolomics data modalities with different deconvolution signal deconvolution tools. So the main tools that we use in my work is independent component analysis. Imagine that you have several sources of signal. You can record them with several microphones, get mixture of signals, and then apply an independent component analysis. You can decompose this mixture of signals into separate signals which will be really close to their initial signals. And this method could be successfully used for different types of biological data. If we go to matrices, imagine that you have matrices with features in rows and samples in columns. Features could be genes and whatever you want, proteins, metabolomics. Then apply an ACA, you decompose these matrices into two. First matrix contains features in rows and components and columns. And here each column represents a dependent signal or independent component that could be annotated with biological processes, cell types, clinical data, and other very useful biological information. And second matrices contain weights for each sample in these new dimensions. And this matrix could be used for prognostics and classification models. And we do this method consensus. That means that we provide robust decomposition, robust independent component analysis. This algorithm was packed into Consica R package, already published in BioConductor. It was surrounded with different functions to provide a full pipeline to data analysis. About data inputs, you can use both bulk and single cell data, combine your small data set with large discovery data set to go deeply what you see what deeply you have in the data, and also apply this method for different data modalities like transcriptomics, epigenomics, proteomics, whatever. And also we added automatic report generation for each component that provide you quick opportunity to quickly understand what hidden signals and hidden dependencies you have in your data. For data integration, we propose to separately deconvolve your data modalities with our ACA, and then using simultaneous analysis, compare weight matrices or the signal matrices to see which dependencies we have. Now moving for some cases, this is a study of melanoma cell lines. We had here 4 NRAS mutant melanoma cell lines, which were treated, and they did single cell RNA sequences before treatment, 1 day after treatment, 4 days after treatment, and 33 days after treatment. And speaking about proliferation rate, after the treatment, it was suppressed, and then 2 cell lines, 33 days after treatment in resistant state had low proliferation rate, and 2 others had a high proliferation rate. This is cells with fast adaptivity to treatment. And then we did metabolomics to try to integrate for this data. So briefly about some interesting dependent components that we extracted. They show expected behavior different for fast and slow adaptivity. So cells with slow, for cells with fast adaptivity, signals go down during the treatment, but after in resistant state, it goes again high, and for cells with slow adaptivity, signals just going down. Another interesting component is a component linked with cell cycle. This is cell division and DNA replication. And if we plot cells in these dimensions, we have such triangles. Cells with slow adaptivity have proliferation cells only before treatment, and cells with fast adaptivity have proliferated cells before treatment and in resistant state. And to be sure this is a real cell cycle, we annotated these cells with several cell cycle stages, and then this is a real cycle granules that we have. Then we did in the lab, our collaborators did metabolomic experiments for the cells, same cell lines. Now we have not a lot of data, only one cell line, and the problem here is it's not really stable. So we have a big variation, and a lot of metabolites have exactly almost the same. So for doing data integration and the comparisons, was really easy, and we're looking for some prediction models, what we can do. And we find published tool, Flux Estimator. They use a network-based model to predict metabolomic fluxes based on RNA-seq data. Here we used TCA cycle network and try to predict metabolic fluxes on our data. And of course, we tried to check the stability of this model. And here was the bad news for us that if we run this tool exactly for the same data, exactly almost the same, we had you see different results. This is like different rates, so it's a question how we can use it. But so of course we try to average them and compare what we have for different stages. For two cell lines that have fast adaptivity, especially mostly for SKM30, we have this again high rates for resistance state. And also we try to compare with our metabolic experiment. Here we don't have as a correspondence with experiment, unfortunately. And also comparing stages for experiments, it's only if you have significance differences between 4 time points. So then we looked for another data set that we can use for prediction models. We find this one with 75 glioblastoma patients. Again, for looking for forward prediction models, mostly all metabolites have really few variation in their data for different patients. And this is just example of prediction models. We ran a couple of them, tried to did on independent components, used features as independent components as genes. And so mostly for, in general, models on independent components works better. And one more project that I want to show you, this is a project contains more than 100 patients, and we had 5 different omics layers here. We apply Constico for each layer separately and then try to find the best components that could predict clinical features. For some of them, mostly, especially for the sex, transcriptomes win. For a couple of them, Metabolomics components win. And in general, we check predictive power of PCA features. Of PCA features, TCA was better. This is a heat map that shows components that have better correlation with factors for different data modalities. Again, some of them for metabolomics and sex for transcriptomics. And then we combine all these components into a correlation-based graph together and add additional notation of factors to understand how these components could be linked together and link with these biological clinical factors. So moving to the summary, we provide a package construct which allows us to deconvolve different data modalities, and for RNA-seq data, we can extract independent signals that could be linked with drug resistance and melanoma. Also, components linked with cell cycle explain faster slope activity of cells. Metabolomics data that we have now show resistance, but not could be really now connected with our transcriptomics data. Prediction of a single cell here model is not stable, but we can suppose of this cycle activation there. SA-based prediction in general gives us better predictions than on genes and PCA-based components, and adding more omics layer increase prediction power. I would like to say thank you for all my lab and other collaborators that were involved in these projects. And thank you for your attention. And we're looking for a new member in our lab, permanent contract for implementation. If you would like to visit Luxembourg, please contact us.
Hey, very good. So time complexes. Let me break the ice. About the convolution that you mentioned, it's reference free. So, but there are nowadays many single data sets that you use for reference. So do you do that to purpose, for better than without reference?
We mean that this is reference free composition. This is unsupervised algorithm. Sometimes, usually, maybe the convolution in bioinformatics means that we do the convolution for cell types, but we use this word in more general sense, like deconvolution of signals. And mathematically, this is called decomposition. Decomposition of matrices, decomposition of signals.
Okay.
Yeah.
Now I get it. You have a question? No question? Then next time again. Ms. Maryna? Simone Montalbano, thanks, Simone, you can start.
Right. So Simone, I'm from the University of Copenhagen, and well, my group focuses mostly on population genetics and disease risk. So it's kind of a bit of a different topic than the rest of it. Of presentation we saw, so I'm going to try to give a introduction and stay on time. Let's see how it goes. So my work is mostly about CVs. Those are structural variants. They consist of deletions and duplication, so a net loss or gain of genetic material in a chromosome. For what we do, we usually define them as between 25 to 50 kilobases to 5 to 10 megabases. So kind of a large and thus usually rare, very rare variants. They have been a classic focus of human genetic research across multiple phases. So CMVs are detected in many different ways for what we do. So very, very large collections of human samples. Actually, genotype data from SNP genotype chips is still the standard. This is because of costs, of course, but also because sequencing data actually for this kind of variation doesn't help us that much in the detection. And regardless of the technology, all methods rely on some measure of the DNA density. This would be the log ratio or the rate depth. Some methods also integrate a measure of the allele composition. Of course, this is doable only if there is a SNP in the marker. For Illumina arrays, this would be the allele frequency. The gold standard software is still considered PenSimV, a quite old software, but it's still the standard. So there are some limitations to it, of course, as you can imagine. This is mostly due to the architecture of the program. So PenSimV uses a needle Markov model to predict the state of each marker based only on the previous one, and then goes on to build larger CMV codes from smaller sets of consecutive SNP with the same predicted copy number, allowing for some missiness in between. As you can imagine, this makes it quite sensible to local noise. Moreover, the DNA intensity, the log ratio, is not stable across the chromosome, even if there is no CMV. So here we see on top, so each dot is a SNP, is a marker, and we see the DNA intensity on top is kind of a normal sample. It's quite stable around 0. On the bottom, you see a very noisy one, and you can see this kind of waviness behavior. This leads to the two main limitations of CMV coding. So one is oversegmentation, so a true code got split in a smaller one, and this is quite easy to solve. And then the second, harder to solve, is false positives. So from our analysis, we know that from 30 to actually more than 60% of PenSimV codes can be false positives. And this is only partially solved by standard filtering. So the only real solution, especially when precision is very important, you can imagine rare variants it is to perform the so-called visual validation, visual inspection. And this is because raw data trends, they take a very visually distinctive pattern. So from the bottom, again, this is the DNA intensity, and you can see it's quite stable across 0, and then it drops because there is a deletion, so there is one less copy. And then this is the allele composition, and the very central mark band is for the heterozygous markers, and you can see there is, of course, a loss of heterozygosity where there is just one copy. So this works well and everything, but however, you can imagine that having a human to go through every single call for a large collection of samples is feasible. And so we thought, okay, why not try to train the computer to do it? This is exactly what we did. So together with my supervisor, I created a data set of almost 16,000 human labeled examples of these true and false CMVs from joint collections of samples from the UK Biobank and from Decogenetics in Iceland. So two different array manufacturers of Affymetrix and Illumina, and a different type of chip array. We also simplified the image for the computer, as you can see here. So again, this is the same image as before, but without all the unnecessary things like axis and stuff. And it's a 96 by 96 pixel grayscale image. And then I trained a relatively simple convolutional neural network with the R implementation of Torch. So it's native R. Then we tested the accuracy in a different set of data within both cohort. And prediction and accuracy are very good. I would say they are well above 90% in both samples. And this is very well comparable to a trained human. The code is, the software is implemented as a formal R package. It's available on GitHub. And we have a paper on bioRxiv coming very, very soon, like last week in theory, but next week, most likely. So that's cool and all. We train the computer to do a very boring task, and it works very well, but what can we actually do with it? So we apply the method to the entirety of the UK Biobank. So we have, after some filtering, just above 500,000 good CMVs from 462,000 samples. And in some way, this is the first time this kind of variation has been characterized on such a large genome-wide and large human collection scale. And this comes with some complexities. So for example, here, I'm showing some simplified segments, some CMVs in blue. And you can see, even if there is no scale, I can tell you that these two are quite similar. They could be the same, exactly the same variant, but then they don't have the same start and stop. So you might want to group them together if you want to have some power to detect something. However, on the right, we can see a more complicated example. So what is the best way to group, to reduce the complexity while retaining as much power and as much specificity as possible is kind of a complex question to ask. We try to do it. I'm not going to cover it today for time's sake, but we try to do something with network analysis and community detection algorithm. If you have some interest on it, let me know. And I'm not going to show that much because actually the easiest way is to forget completely about the segment structure and use regular beans or windows across the genome. And then each bean is going to be the marker and each sample is going to be a carrier if they have a CMV overlap in the bean or not. And of course, you can also use whatever as bean. So yeah, genes and whatever you want. And this is kind of what I'm showing here. So it's kind of full, it's a very filled figure, but we are on the Q arm, chromosome 22. So it's a little bit zoomed in. And if we focus just on the red plot, it's a line plot of beans, regular beans, 250 kilobase across the genome. And then the value is the log 10 n, where n is the number of CMVs in that bean. And we can see that pretty much the entirety of the chromosome is covered by at least some. But we can also clearly see that there are some very precise outposts or some larger places where there are much more CMVs than the rest of the genome. And we can ask ourselves, is this what we would expect or not? We try to answer this by setting kind of a baseline. So we simulate, we shuffle CMVs around 1,000 times in a clever way. So we try, this is the SNP map. We follow the SNP map because of course we couldn't ever detect anything if that wasn't a marker. But then we also try in some ways to preserve this kind of hotspots behavior that the real distributions have. And we shuffle them around 1,000 times, and then we take the median for each bean, and that builds our baseline. Then we can compare the baseline with the real, and that we get a compute a log twofold change and a Fisher test p-value. And here I'm showing the significant hits deletions in red and duplications in blue. So we can see already from here, we can see a lot of things, right? So we can see that actually there are some places where you have way more deletions or duplications than expected, and there are some places where you have less. And of course, you can start to ponder why. Is there some recurrency in these, some segregating element, or is there some protected genes or something? And even more, you can start to ponder what's going on here. Like that region is protected against deletion, but not against duplication. And I mean, you can have a lot of questions, and this is what I'm working on right now. But I'm not gonna present much more than that. So thank you for your attention. Thank you for, thanks to my institute, my supervisor Andres, the director Thomas, and Brian at the Deeper Genetics in Iceland. And thanks for the attention.
Thanks for being time. Have questions?
Thank you very much. Very great talk. Really love the topic of copy numbers. Close to my heart. And I really applaud the approach of actually validating them computationally, not just by eye. With regards to training of your network, have you taken measures to avoid learning biology? Because we know that not all copy numbers, so there is a biological structure behind copy number variation. And I just wonder what you did to avoid that.
Yeah, okay, so the easiest, yes, in some ways, yes, in some ways, no. So what we did is we used multiple different arrays with different coverage across the genome, and that already kind of protects us in a way. But then something I didn't show is that in the UK Biobank part of the training set, which is the largest one by far, I actually downsampled very frequent region, exactly for that reason. So to avoid that the model would train on some specific genomic loci present only in that specific array and in that specific population. Of course, you can do much more than this. But yeah, and then in another way, in the deeper set, actually, we are using trios. So also there, we have kind of a multi-layered internal validation in a way, because you can look at the entire trio at the same time. And but just to summarize, I wouldn't really exclude too much, because otherwise you are actually doing the opposite of what you want to do, right? So in some ways, yes, and that's what we did, but in some other ways, no.
Just follow-up question to that. Have you tried shuttling along the x-axis? Because if you look at those plots, something what people might not know is that essentially you go along the x-axis, you go along the chromosomes.
Yeah.
So I wonder if you just train essentially chromosome Y losses and cancer frequency of this lesion, for example. And have you thought about shuttling those along the x-axis?
Yeah. Okay. So there is a very naive implementation of that, which is data augmentation by just flipping the image and the Y. That's something we do. And something I wanted to do is actually, I think, what you're suggesting. So taking the true deletions and moving it somewhere else, right?
Actually, just shuttling the chromosomes, I think, would be enough.
Yeah. Yeah. No, I'm not doing it. But that's something we can do. But so I trained a lot of models, of course. This one I'm happy with. It's working fine. But of course, there is no limit to how much things you can do, right? So at the moment, I stopped training because it's good enough for what we do. But the accuracy we tested kind of within sample in a way, even though we are training mostly on the UK Biobank and we are testing mostly on Decode, it's still kind of within sample. And so if we want to make it really sample agnostic, then probably we would need to do something similar to what you suggested. Thank you.
Great discussion. So we're saying Simone again.
Hi everyone, and thanks for this wonderful congress. And as the last speaker, I will present something a bit different. So about my work on Pedixplorer package dedicated to manage, filter, and visualize pedigree data. So pedigree data are a structured way to represent family history, specifically focusing on how traits, genetic variants, or disease are passed down through generation. Therefore, to draw a pedigree, you need a lot of different information for each individual. You need to know his sex, if he's affected, still alive. Thanks. And how he is related to other individuals. It can be a spouse, a child, or a twin. You also need to know how each affection and traits needs to be represented in the pedigree for filling, bordering, shades, and how to align each individual in regard of each other's. The first convention to standardize how the pedigree drawing was written in 1995. They described how each symbols, relationships, and other information needs to be represented. Pedigrees are used in various fields. In animal breedings, they offer a detailed record of an animal's ancestry, helping breeders make informed decision to enhance desirable traits, manage genetic diversity, and while reducing the risk of inherited disease. In genetic counseling, pedigrees are used to to detect to manage the occurrence of inherited condition by tracing the occurrence of genetic traits across multiple generations within a family. Finally, pedigrees are also used in genomic studies to infer the inheritance and transmission mode of disease. Based on them, it is possible to hypothesize if a disease is dominant or recessive, autosomal, or linked to the X chromosome. They also offer useful information to select individuals with lower kinship for genome-wide association studies and individuals from informative families for linkage analysis. However, it isn't always as simple. For pets and livestock, pedigrees are really much more complex than those found in human studies and can be exceptionally large. Moreover, to select traits of interest, breeders often induce founder effects where one individual participate a lot to the next generation. This results in a lot more inbreeding and generation mixing, complexifying a lot the pedigree visualization. Therefore, how can we easily manage such complex data and visualize only the relevant information? To do so, many different informatics solutions already exist, more or less automatized and maintained. Proprietary softwares are often quite expensive, and open source one doesn't well handle the huge pedigrees. Cranefoot and Madeline, on the other hand, it works really well, but they are complicated to install and lacks functionalities. Finally, Kinship 2 was the main package in R available to draw pedigrees. It worked well, but wasn't maintained due to the author's lack of time. Therefore, as I needed an enhancement for my PhD thesis, I've decided with the authors to create a new implementation of the package inside the BioConductor community. The Kinship 2 code needed to be reorganized with more customization, documentation, and unit test. New function to query huge pedigrees was also necessary, as well as an easy way to use all the main function. For today, I would like to focus on 3 of the improvements made. The S4 classes to represent pedigree data and to allowing a better type checking and structured object definition. Second, the new function and organization of the package. And finally, the Shiny application. The new S4 object for pedigrees is organized as follows. the main class called PED, store the individual information as an S4 object with parallel slot. The rel class, on the other hand, contains the special relationship that cannot be easily stored in the PED class, such as twins or spouse with no children. The scale class has two data frames, one for the filling and the other one for the ordering of the symbols. And finally, the ins class enabled to specify the relative horizontal ordering between siblings and between spouses. All the main functions have been rewritten as methods to use the S4 class. Preprocessing the function have been added to simplify the process, to fix common errors and normalize the data. The constructors are then used to generate a pedigree object that can be then used to compute and extract more information. This allows to filter and shrink the pedigree before aligning the individuals before plotting. With all these new classes and functions, it might be complicated for a new user to know what to do at which point. Therefore, I developed a Shiny application to use all the main functions. As inputs, you only need a file with the ID of the individuals, his father, mother, and his sex, only mandatory. It is accessible locally through a pet Shiny function and also on a web server on a virtual machine. As the application is quite complex, I've decided to work with Shiny models. The principle is simple. You create small standalone app dedicated to only one task. They therefore have a server function, a user interface, a small demo, and that allows to unit test all of them separately to ensure the reliability of each step before testing the whole app. The app looks as follows. The workflow is first to import the data for a different file, and after you can select the health option and the modality of affected individuals. You can then select and change the color, and their family information are then displayed. You can sub-select and filter the pedigree from different individuals, and then select one of the resulting subfamily. You can render the plot interactively to have more information on a given individuals or to zoom on a specific part of the pedigree. You can then download the pedigree in different formats. To summarize, the Pedixplorer package enable a better control over pedigree data using S4 object. The computational load is reduced for each pedigree, and additional function enabled to get the relevant information. Finally, the Shiny application make it easy for new user to use all the main function. I would like to thank my team members, the original authors of the Kinship 2 package, as well as the guide dogs of organization financing my thesis. And thanks you for your attention, and I'm happy to answer any question.
Great, so good in time. Questions? I haven't mentioned, but I guess that it includes a parser for pet files package and read pet files.
Yeah. Which package is that?
Your package can read pet files.
Yeah, that's one of the aims, to be able to use any kind of data and import them directly to create a common S4 package for any, because there exist other packages such as Pet Tools from AG that use the Kinship 2 package, but is now deprecated. We need to have, I think, common S4 object for everyone to use and to have a converter to use classical input.
Okay. Question?
I'm not gonna talk about pedigree and make a fool of myself, but I'd like to take the opportunity to invite you, if you wanted to, the BioConductor contribution guide website where we try to help people develop their package. There is a Shiny page to that still doesn't include any guidance for Shiny modules. which come again and again in submissions. People like to use it. I can't help it. I like Shiny, but I've never used modules. So I'd like to invite you if you wanted to essentially contribute a section over there.
Yeah, I'm on the Slack channel on that, and yeah, there is a lot of tweaking to be done for the unit test of Shiny modules, but because it's not natively supported in the Shiny test 2 package, so it's a bit complicated, but it's doable. And so yeah, maybe we should have a bit more guidelines on that to make it easy for all the developers.
Yeah, so that would be very helpful. And the guidelines are open for everyone to contribute. Some people actually take them at sort of Bible face value, saying, oh, this is the truth, I need to follow it. But I do keep emphasizing on the Shiny side that I wrote most of them and the way I saw Shiny, but I know that modules are there, I'm just not using them. And so that's where every time I see that, I'm like, I would love for someone to write it and then I can learn from them.
Yeah, it's really useful to separate each task, make it, you do not have only one big server file with thousands of lines. So that's helpful when you want to debug.
Okay, thanks. Let's thank again and all the speakers of the session. Now time for the poster pitches of the second poster session. So those of you
Hi, everyone. So I'm going to be presenting a pipeline for DAISY-based mass spec imaging, wherein I use various packages from BioConductor for exploration, peak selection, and processing, and then annotate using especially the packages for mass spectrometry, and then finally confirm them with MS2 confirmation. So this is just an example of how the data would look like. You would have an HAE staining and how the different organs can be clustered. And the overlay is not displaying properly here, and then finally I'm developing this as a Shiny app to help users as a graphical user interface as well. Thank you.
So this is a poster about Athena, former student we talk about today at Athena Awards. This is a package for analysis of transposable elements. You know that they are mobile genetic elements that are repetitive and interspersed on the genome, which makes them difficult if you want to quantify the expression of them, because they basically multi-mapping reads go to those locations. And this package facilitates integration of annotations and their expression quantification into BioConductor workflows for analysis of RNA-seq data by using a common interface for 4 different methods that are implemented in the package. That's it.
Hi, is it on? Okay. Hi, so I'm Sarah. I'm presenting Center. It's a BioConductor package for cell type specific enhancer target prediction. We want to submit it soon. It's a bit more general than the crop enhancer target step. And it's also more fine-tuned and it looks at more cases that could happen. I'm not going to go over again about the enhancer target stuff because Persia explained it so well. So yeah, if you're interested in that, we use minimal data in our model and we use very routine experiments to give predictions that are as good as some of the state-of-the-art models. And yeah, so come by if you're interested in that. Also, if you're someone who knows about how to implement annotation hub packages, please come by because I would love to talk to you. Thank you.
Good afternoon, everyone. I am Elena. My poster is focused on a benchmark of single-cell batch effect correction and the creation of our package called BatchChef. This package is available on GitHub and contains a common interface for single-cell batch correction method in R and in Python environments. If you are interested or you have any feedback, come to see my posters.
Hello, everyone. I'm Himmi, and I'm going to talk about a human gut microbiota that is a complex and dynamic ecosystem.
but how can we uncover its key ecological characteristics that associate with health outcomes? Our research aims to answer this question by applying unsupervised learning techniques to identify microbiota community types within a large Finnish population cohort data. We utilized a MIA package for microbiome analysis tools and in a tree summarized experiment container. And after identifying these community types, we analyzed their association with the mortality endpoint obtained for Finnish health registers within almost 18 years follow-up. And we actually were able to detect the community type that associates with early mortality. So if you got interested about this, come to see my poster. Thanks.
Hello, everyone. So my poster is about the trajectory-based differential expression analysis with MS-Group2. So we could already do complex designs with MS-Group2. We have now extended it, so you can also do the trajectory analysis or time course data analysis. And in my poster, I'm showing how we can do this on the AML dataset from SCOOP, which is quite well known, where you see that you have this trajectory of the stem cells that go to progenitor and blast cells. And this you can easily put into the model as a smoother, which you can then have the interesting part, which is the inference, where you have different hypothesis tests, where you can see like, oh, these are the same shape, but different location. Or you can find proteins with different shape. And maybe these are also kind of like shift in location, but you could also have proteins with the same location. And you can also do clustering, where you can find proteins that are the same shape. So if you're interested in that, come say hi to the poster.
Hi everyone, I'm Lotta, and I'm wondering if you also sometimes struggle going shopping, like going from one shop to the other to get everything that you need, like Kim Kardashian over there. In our project, we're not keeping up with the Kardashians, but we're keeping up with protein-protein interaction databases. And if you want to work with PPI data, currently you have to go from one database to the other, and that can be quite nasty. So what if I've told you that there will be a package called Network Hub that provides you with a graph of interactions, and if you have a summarized experiment data frame, you can plot your data on that graph. And if you want to keep up, come to my poster. I'm happy to talk to you about it.
Hi, I'm Leo, and we are developing a whole ecosystem of techniques for microbiome research. This includes tools for data. We have techniques for data access from public databases, including curated metagenomic data, hollow food, and EBI magnified database. And this is all multi-omics data, so we've fetched it into three summarized experimental multi-assay experiment formats supporting those. We have ecosystem of techniques to analyze and visualize microbiome data, also IC3 extension for interactive analytics, and then reproducible documentation in the form of a Git book orchestrating microbiome analysis. So if you're interested in microbiome research, just come to the poster, and Thomas will give a tutorial tomorrow. Thank you.
Hi, everyone. As you can see, I'm not Leon. That's a pity, because first of all, you don't get to meet a talented bioinformatician that was working with us in the group. And the second, the worst part is that you have to bear with me. So bear with me in a long way. I'll count with you. Leon had one task. He needed to go through the packages that we had and try to modularize, factorize, and rework on the function that were in there. He came up with two objects that he wanted to work with, a DE container and the result of differential expression analysis, which is something that we have to do commonly. He had three directions that he wanted to go through, so obtaining details and overviews, sliding smoothly into the downstream analysis, and decorating reports at best. I would also have, so here, a small aperitif of what you would get, and I would also count to 4, but Fabrizio actually stole my fun for the 4, so you could see something that comes from the 4th best university after Oxford, Cambridge, and Louvain, and that order. I can also count until 41 today, but if you're interested into seeing this, come to see our poster. Thank you.
Hello, everyone. I'm Mahesh. This is my poster. This is work done on pan cancer based on TCG dataset. since this was a public data set, it was easy for us to identify where invasiveness is a significant feature or kind of prognostic in the set of 32 cancers. And we essentially pre-processed our data, use a classifier for low, medium, and high invasiveness. We used RGBM, which is regularized Boltzmann machine, to essentially find out the transcription regulators and the top master regulators. So these transcription factors that we found out were then sorted the top 10 into top master regulators. And then we visualized them and we did pathway enrichment analysis. This can be used to target them and make drugs out of the enriched pathways. Again, as for yesterday, my world has been shattered by all models are wrong lecture. So I am still pursuing a PhD in translation medicine, and I'm really questioning myself. So thank you so much. Please do not give in to my existential crisis, and please come and visit my poster. Thank you.
Okay, let's conclude this session. Today I will present you one of my PhD projects, that is the development of Samurai, an XLO-based pipeline for the analysis of copy number from shallow whole genome sequencing data. So very briefly, you have the pre-process step, the copy number analysis block, and then a post-processing step, sorry. And we included very cool modules in the copy number block. So if you're curious, come to see my poster and let's discuss together. Thank you.
Thanks to all the speakers on the poster pitch. So now the poster session starts. And remember that at 6 o'clock, our very local organizer, Ollie group, is going to give us a tour through Oxford.